ಏನು ಕೊಡ್ತೀನಿ ಸರ್ ಈಗ.
Okay. Very good evening. ಎಲ್ಲರಿಗೂ ಶುಭ ಸಂಜೆ. I welcome today's speaker, Dr. N. Samba Kumar sir, on behalf of KSTA, on behalf of all the students and teachers who are with us now, and on my own behalf. Hearty welcome to you, sir.
Thank you.
And thank you very much for accepting our invitation to deliver this webinar in this program of Talent Search program. Also, I would like to welcome today's teachers and students who are with us in this WebEx as well as YouTube platform, and my colleagues and other team members. Before starting today's session, I would like to give a brief introduction about Dr. N. Samba Kumar sir. Sir has specialization in developing field protocols for surveying large mammals in Asian forests, and his research focuses on evaluating factors that shape distribution and abundance of large mammals. His field research work included reliably estimating populations of tiger, its co-predators, and prey in different biogeographic habitats within India at multiple spatial scales and monitoring their populations over time. He has authored 2 books and more than 85 scientific articles, technical reports, and book chapters. Samba Kumar sir has taught field survey techniques for monitoring large mammal populations to a large number of wildlife enthusiasts that include Forest Department field protection staff, aspiring wildlife biologists, and amateur naturalists. He has conducted more than 100 field training workshops over past 25 years and inspired generations of volunteers to pursue wildlife research and conservation. As an honorary director with the Wildlife Preservation Group, which is known as WILPEG, a non-governmental organization, Samba Kumar sir has conducted more than 100 nature camps and wildlife education or awareness programs to the students, teachers, rural youth, and urban naturalists. Prior to pursuing his career in wildlife biology, Samba Kumar sir served in URSC ISRO Satellite Center of Government of India for 14 years, during which he imparted nature and wildlife education to hundreds of ISRO employees through nature walks and field trips. Sir has served with Center for Wildlife Studies from 1994. In 2016, he relinquished his position as the additional director for Science and Training at India program of the Wildlife Conservation Society, which is known as WCS, an international NGO based in New York. He is currently an emeritus scientist at the Center for Wildlife Studies. He received his graduate degree in management from University of Mysore, postgraduate degree in ecology from Pondicherry University, and PhD from Manipal University. He was conferred with the prestigious Cow Jess Wildlife Conservation Award for the year 2010 in recognition of his outstanding services, a certificate of merit in 2006 by New York-based Wildlife Conservation Society, and several other awards and honors from local and regional associations. With this brief introduction, I once again welcome Dr. N. Samba Kumar sir to deliver today's session. Thank you, sir. Over to you, sir.
Thank you, Umesh. Thank you for the nice introduction. I feel happy to talk to the students about a topic which is very hot in actually today's, in recent news. ನೀವು ಇತ್ತೀಚೆಗೆ ಬಹಳ ಸುದ್ದಿ ಮಾಧ್ಯಮದಲ್ಲಿ ಕೇಳಿದ್ದೀರಾ, ಭಾರತದಲ್ಲಿ ಹುಲಿಗಳ ಸಂಖ್ಯೆ ಜಾಸ್ತಿ ಆಗಿದೆ, ಚಿರತೆಗಳ ಸಂಖ್ಯೆ ಬಗ್ಗೆ, ಮತ್ತೆ ಇತ್ತೀಚೆಗೆ ಆನೆಗಳ ಸಂಖ್ಯೆ ಬಗ್ಗೆ ಕೂಡ ಬಹಳ ದೊಡ್ಡ ಸುದ್ದಿಯಾಗಿತ್ತು. ನಮ್ಮ ಕರ್ನಾಟಕ ಹುಲಿಗಳ ಸಂಖ್ಯೆಯಲ್ಲಿ 2ನೇ ಸ್ಥಾನದಲ್ಲಿದೆ, ಮಧ್ಯಪ್ರದೇಶದ ನಂತರ. ಈ ಥರ ವನ್ಯಪ್ರಾಣಿ ಸಂಖ್ಯೆ ಬಗ್ಗೆ ಬಹಳಷ್ಟು ತೀವ್ರವಾದ ಗಮನ ಇತ್ತೀಚೆಗೆ ಬಂದಿದೆ. ಸೊ ನಾನು ಯೋಚನೆ ಮಾಡಿದೆ, ಇಂಥ ಒಂದು ಸಂದರ್ಭದಲ್ಲಿ ನಿಮ್ಮಲ್ಲಿ ಬಹಳಷ್ಟು ಜನರಿಗೆ ಒಂದು ಕುತೂಹಲ ಮೂಡಿರಬಹುದು. ಇದು ಹೇಗೆ ವನ್ಯಜೀವಿಗಳ ಸಂಖ್ಯೆ ಗಣತಿಯಾಗುತ್ತೆ, ಇದು ಎಷ್ಟರ ಮಟ್ಟಿಗೆ ನಿಖರವಾಗಿದೆ, ಹಾಗೂ ಇದರ ಹಿಂದಿರುವ ವಿಜ್ಞಾನ ಏನು, ವೈಜ್ಞಾನಿಕ ವಿಧಿವಿಧಾನಗಳ ಬಗ್ಗೆ ನಿಮಗೆ ಒಂದು ಕಿರುಪರಿಚಯ ಮಾಡಿಕೊಡುವ ಪ್ರಯತ್ನ ಇವತ್ತು ಮಾಡ್ತೀನಿ. ನಾನು ಒಂದಷ್ಟು ಪವರ್ ಪಾಯಿಂಟ್ ಸ್ಲೈಡ್ಸ್ ಹಾಕಿದ್ದೀನಿ. ಅದಲ್ಲದೆ ನಾನು ಅನ್ಕೊಂಡೆ ಮಾತ್ರ ಅಲ್ಲದೆ ಒಂದು ಪುಟ್ಟ ವಿಡಿಯೋ ಮೂಲಕನೂ ಕೂಡ ಈ ವನ್ಯಜೀವಿ ಗಣತಿ ವಿಧಾನವನ್ನ ಫೀಲ್ಡ್ ಅಲ್ಲಿ ಹೇಗೆ ಮಾಡ್ತಾರೆ ಅನ್ನೋದನ್ನು ಕೂಡ ನಿಮಗೆ ತೋರಿಸ್ಬೋದು ಅಂತ ಒಂದು ಆಲೋಚನೆ ಬಂತು. ಇದು ಈ ವೆಬ್ಎಕ್ಸ್ ಟೆಕ್ನಾಲಜಿ ನನಗೆ, ಅಂದ್ರೆ ನಾನು ಈ ವಿಡಿಯೋ ಎಲ್ಲ ತೋರಿಸೋದು ಮಾಡೋದು ಅದರೆಲ್ಲ ನಾನು ಬಹಳಷ್ಟು ಪರಿಣಿತಿ ಇಲ್ಲ. ಬಟ್ ನಾವೊಂದು ಈಗ ಟ್ರಯಲ್ ಮಾಡಿ ನೋಡಿದ್ವಿ. ಅದು ಸಾಧ್ಯ ಆದರೆ ವಿಡಿಯೋ ಕೂಡ ತೋರಿಸ್ತೀನಿ, ಇಲ್ಲ ಅಂತಂದ್ರೆ ಪವರ್ ಪಾಯಿಂಟ್ ಪ್ರೆಸೆಂಟೇಷನ್ ಅಂತೂ ಖಂಡಿತ ಸಾಧ್ಯವಾಗುತ್ತೆ. ಎಲ್ಲರಿಗೂ ಕಾಣ್ತಾ ಇದೆ ಅನ್ಕೋತೀನಲ್ಲ. ಸೊ ಇಂದಿನ ವಿಷಯ ವನ್ಯಜೀವಿ ಗಣತಿ ಬಗ್ಗೆ. ಹಿಯರ್ ಎ ಟೈಗರ್, ದೇರ್ ಎ ಡಿಯರ್, ಹೌ ಟು ಕೌಂಟ್ ಎನಿಮಲ್ಸ್ ರಿಲಯಬಲಿ ಇನ್ ದಿ ವೈಲ್ಡ್. ಇದು ರಿಲಯಬಲಿ ಅನ್ನೋದು ಬಹಳ ಮುಖ್ಯವಾದ ಶಬ್ದ. ಅಂದ್ರೆ ನಂಬಲರ್ಹವಾದ, ವಿಶ್ವಾಸಾರ್ಹವಾದ ಗಣತಿಯನ್ನ ಹೇಗೆ ಮಾಡುವುದು. ಯಾಕಂದ್ರೆ ಸುಮ್ಮನೆ ಒಂದು ಅಂದಾಜು ಮೇಲೆ ಗುಂಡು ಹೊಡೆಯೋದು ಬೇರೆ, ವೈಜ್ಞಾನಿಕ ವಿಧಿವಿಧಾನದಲ್ಲಿ ವನ್ಯಜೀವಿ ಗಣತಿಯನ್ನ ನಡೆಸೋದು ಬೇರೆ. ಇದರ ಬಗ್ಗೆ ಬಹಳಷ್ಟು ಕಡಿಮೆ ಮಾಹಿತಿ ಸಾಮಾನ್ಯ ಜನರಲ್ಲಿ ಇದೆ. ಸೊ ಇದು ಹೇಗೆ ಮಾಡ್ತಾರೆ ಅನ್ನೋದ್ರ ಬಗ್ಗೆ ಇವತ್ತು ತಿಳ್ಕೊಳ್ಳೋಣ. ಈಗ ವನ್ಯಜೀವಿ ಅಂದ ತಕ್ಷಣ ಎಲ್ಲರ ಮನಸ್ಸಿನಲ್ಲಿ ಮೊದಲಾಗಿ ಬರೋದು, ಅದರಲ್ಲೂ ನಮ್ಮ ಭಾರತದಲ್ಲಿ ಹುಲಿ ಇರ್ಬೋದು, ಚಿರತೆ, ಸೀಳು ನಾಯಿಗಳು. ನಿಮ್ಮಲ್ಲಿ ಎಷ್ಟು ಜನ ಇದನ್ನ ಕಾಡಿಗೆ ಹೋಗಿ ನೋಡಿದಿರ ನನಗೆ ಗೊತ್ತಿಲ್ಲ. ಇವೆಲ್ಲ ಮಾಂಸಾಹಾರಿ ಪ್ರಾಣಿಗಳು. ಇವು ಕಾಣಸಿಗಲಿಕ್ಕೆ ಬಹಳ ಅಪರೂಪ. ಮತ್ತೆ ಹುಲಿ, ಚಿರತೆ ಆ ತರ ಮಾರ್ಜಾಲ ಜಾತಿ ಪ್ರಾಣಿಗಳಾದ್ರೆ ಅವು ನಿಶಾಚರಿಗಳು, ರಾತ್ರಿ ಹೊತ್ತು ಬಹಳಷ್ಟು ಸಂಚರಿಸುವುದು. ದೇ ಆರ್ ಸೀಕ್ರೆಟಿವ್ ಎನಿಮಲ್ಸ್, ನೋಡ್ಲಿಕ್ಕೆ ಕಷ್ಟ, ಅಂಥದ್ರಲ್ಲಿ ಗಣತಿ ಮಾಡೋದು ಹೇಗೆ? ಈ ತರ ಒಂದು ಪ್ರಶ್ನೆಗಳು ನಿಮ್ಮಲ್ಲಿ ಉದ್ಭವ ಆಗ್ಬೋದು. ಕರಡಿ, ಆನೆಗಳು, ಸ್ಪಾಟೆಡ್ ಡಿಯರ್, ಚೀತಲ್, ಕಡವೆ, ಸಾಂಬಾರ್ ಅಂತ ಹೇಳ್ತಾರೆ, ಕಾಟಿ, ಗೌರ್ ಅಂತ ಹೇಳ್ತಾರೆ ಇದರಲ್ಲಿ. ಇದು ಬಾರ್ಕಿಂಗ್ ಡಿಯರ್ ಅಥವಾ ಮಂಟ್ ಜಾಕ್, ಕಾಡು ಕುರಿ. ಸೊ ಈ ಪ್ರಾಣಿಗಳು, ಈ ಸಸ್ಯಾಹಾರಿ ಪ್ರಾಣಿಗಳು ಬಹಳಷ್ಟು ದಿನದಲ್ಲಿ ಆಕ್ಟಿವ್ ಆಗಿರುತ್ತೆ. ಡಯೂರ್ನಲ್ ಎನಿಮಲ್ಸ್ ಅಂತ ಕರಿತೀವಿ. ಮತ್ತೆ ಕಾಡಿನಲ್ಲಿ ಹೋಗ್ತಾ ಇರಬೇಕಾದ್ರೆ ಇದು ಮಾಂಸಾಹಾರಿ ಪ್ರಾಣಿಗಳಿಗೆ ಹೋಲಿಕೆ ಮಾಡಿದ್ರೆ ಬಹಳ ಈಸಿಯಾಗಿ ನೋಡ್ಲಿಕ್ಕೆ ಸಿಗುವಂತ ಪ್ರಾಣಿಗಳು. ಇವು ಓಡುತ್ತವೆ. ಆದ್ರಿಂದ ನಾವು ಕಾಡಿನಲ್ಲಿ ಹೋಗ್ತಾ ಇರಬೇಕಾದ್ರೆ ಎಷ್ಟೋ ಸಲ ನಮಗೆ ಕೆಲವು ಸಲ ಕಾಣ್ಲಿಕ್ಕೆ ಸಿಗುತ್ತೆ, ಬಹಳಷ್ಟು ಸಲ ಕಾಣ್ಲಿಕ್ಕೆ ಸಿಗಲ್ಲ. ಸೊ ಈ ತರ ಒಂದು ಪ್ರಾಣಿಗಳು, ಸಸ್ಯಾಹಾರಿ ಪ್ರಾಣಿಗಳು ಹೇಗೆ ನಾವು ಗಣತಿ ಮಾಡೋದು? ಅದಲ್ಲದೆ ವನ್ಯಜೀವಿ ಬಹಳ ಬೇರೆ ಬೇರೆ ಪಂಗಡಗಳೆಲ್ಲ ಉಂಟು. ನೀವು ಇನ್ಸೆಕ್ಟ್ಸ್ ಕೇಳಿದಿರ, ಚಿಟ್ಟೆಗಳು, ಕಪ್ಪೆಗಳು, ಪಕ್ಷಿಗಳು. ಸೊ ಈ ತರ ಬೇರೆ ಬೇರೆ ಸಮೂಹ, ಪ್ರಾಣಿಗಳ ಸಮೂಹ ಅಥವಾ ಜಾತಿಗಳಿಗೆ ಬೇರೆ ಬೇರೆ ಒಂದು ರೀತಿಯ ವಿಧಿವಿಧಾನಗಳನ್ನ ಅನ್ವಯ ಮಾಡಬೇಕಾಗತ್ತೆ. ಈ ದಿನ ನಾವು ಪ್ರಮುಖವಾಗಿ ಮಾತಾಡೋದು ಲಾರ್ಜ್ ಮ್ಯಾಮಲ್ಸ್, ಅಂದ್ರೆ ದೊಡ್ಡ ಸಸ್ತನಿ ಪ್ರಾಣಿಗಳು. ಅದರಲ್ಲೂ ಎರಡು ಬಹಳ ಮುಖ್ಯವಾದ ಪಂಗಡಗಳು ಅಂತಂದ್ರೆ ಮಾಂಸಾಹಾರಿ ಪ್ರಾಣಿಗಳು ಮತ್ತೆ ಸಸ್ಯಾಹಾರಿ ಪ್ರಾಣಿಗಳು. ಇವೆರಡರ ಇವೆರಡರ ಬಗ್ಗೆ ಸ್ವಲ್ಪ ಜಾಸ್ತಿ ಫೋಕಸ್ ಮಾಡಿ ಯಾವ ಪದ್ಧತಿ, ಯಾವ ವೈಜ್ಞಾನಿಕ ವಿಧಿವಿಧಾನಗಳನ್ನ ನಾವು ಅನುಸರಿಸಬಹುದು, ಇವನ್ನ ಬಹಳ ಅಕ್ಯುರೇಟ್ ಆಗಿ ರಿಲಯಬಲ್ ಆಗಿ ಗಣತಿ ಮಾಡಬೇಕು ಅಂದ್ರೆ ಅನ್ನೋದನ್ನ ತಿಳ್ಕೊಳ್ಳುವ. ಈ ವಿಧಿವಿಧಾನಗಳ ಇದು ಕಾಡ್ಹಂದಿ ಅಲ್ಲ, ಇದು ಕೂಡ ಬಹಳ ಸಲೀಸಾಗಿ ಸಿಗುವಂತ ಪ್ರಾಣಿ. ಈಗ ಇದನ್ನ ನಾವು ವನ್ಯಜೀವಿ ಗಣತಿ ವಿಧಾನದ ಬಗ್ಗೆ ಮಾತಾಡೋಕ್ಕಿಂತ ಸ್ವಲ್ಪ ಮುಂಚೆ ನಾನು ಒಂದು ಸಂಕ್ಷಿಪ್ತವಾಗಿ ಹೇಳಬೇಕು ಅಂದ್ರೆ, ಈ ಪ್ರಾಣಿಗಳ ಸಂಖ್ಯೆ ನಿಖರವಾಗಿ ಅಂದಾಜು ಮಾಡೋದು ಬಹಳ ಮುಖ್ಯ. ಅದಕ್ಕೆ ಕಾರಣಗಳು ಹಲವಾರು ಉಂಟು. ಒಂದು, ನಾವು ವನ್ಯಜೀವಿ ಸಂರಕ್ಷಣೆ ಮಾಡಬೇಕು ಅಂದಾಗ ಈಗಿನ ಸ್ಥಿತಿ ಹೇಗೆ, ಯಾವುದೇ ಒಂದು ಪ್ರದೇಶದಲ್ಲಿ ಪ್ರಾಣಿಗಳ ಸಂಖ್ಯೆ ಸಮೃದ್ಧವಾಗಿದೆಯೇ, ಮತ್ತೆ ಅದನ್ನು ನಿರ್ವಹಣೆ ಮಾಡಲಿಕ್ಕೆ ಉಪಯೋಗಿಸೋ ಮ್ಯಾನೇಜ್ಮೆಂಟ್ ಆಕ್ಷನ್ಸು ಅಥವಾ ಸಂರಕ್ಷಣಾ ವಿಧಾನಗಳೇನಿದೆ ಅವು ಪರಿಣಾಮಕಾರಿಯಾಗಿದೆಯೇ. ಅವು ಪರಿಣಾಮಕಾರಿಯಾಗಿ ಇದ್ರೆ ವೈಲ್ಡ್ ಲೈಫ್ ಪಾಪ್ಯುಲೇಷನ್ಸ್ ವಿಲ್ ಬಿ ಇನ್ ಗುಡ್ ಹೆಲ್ತ್, ವಿಲ್ ಬಿ ಇನ್ ಗುಡ್ ನಂಬರ್ಸ್. ಹಾಗೆ ನಮ್ಮಲ್ಲಿ ಒಂದಷ್ಟು ಮೂಲ ಉದ್ದೇಶಗಳಿರುತ್ತೆ. ಈಗ ಟೈಗರ್ ರಿಸರ್ವ್ಸ್ ಅಂದಾಗ ಅದರಲ್ಲಿ ಮೂಲ ಉದ್ದೇಶ ಏನು? ಹುಲಿಗಳ ಸಂಖ್ಯೆಯನ್ನ ಆ ಪ್ರದೇಶಕ್ಕೆ ಸಾಮರ್ಥ್ಯದ ಅನುಗುಣವಾಗಿ ನಾವು ಎಷ್ಟಿದೆ ಅಂತ ಮೇಂಟೈನ್ ಮಾಡ್ಕೊಂಡು ಬರೋದು, ಕಮ್ಮಿ ಇದ್ದರೆ ಜಾಸ್ತಿ ಮಾಡೋದು, ಅಥವಾ ಹೇಗೆ ನಾವು ಎಷ್ಟರ ಮಟ್ಟಿಗೆ ಸಮೃದ್ಧವಾಗಿ ನಿರ್ವಹಣೆ ಮಾಡ್ತೀವಿ ಅನ್ನೋದು, ಈ ತರ ಉದ್ದೇಶಗಳಿರುತ್ತೆ. ಆ ಉದ್ದೇಶಗಳನ್ನ ನಾವು ಎಷ್ಟರ ಮಟ್ಟಿಗೆ ನಿಭಾಯಿಸ್ತಾ ಇದ್ದೀವಿ? Are we succeeding in meeting those objectives, or are we failing in those objectives because of the problems that we are facing? ಇದನ್ನು ತಿಳ್ಕೊಳ್ಳಲಿಕ್ಕೆ ಕೂಡ ನಾವು ಪ್ರಾಣಿಗಳ ಸಂಖ್ಯೆ ಎಷ್ಟು ಅನ್ನೋದು ನಿಖರವಾಗಿ ಅಂದಾಜು ಮಾಡೋದು ಬಹಳ ಮುಖ್ಯವಾಗುತ್ತೆ. ಅದಲ್ಲದೆ ನಾವು ಎಷ್ಟೋ ಸಾರಿ ಬೇರೆ 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 ಸಂರಕ್ಷಣಾ ವಿಧಾನಗಳನ್ನು ಅನುಸರಿಸುತ್ತೀವಿ. ಎಷ್ಟರ ಮಟ್ಟಿಗೆ ಈ ವಿಧಾನಗಳು ಫಲಕಾರಿಯಾಗಿವೆ, ನಾವು ಬೇರೆ ಏನು ಮಾಡಬಹುದು, ಇನ್ನೂ ಇದನ್ನ ಪ್ರಾಣಿಗಳ ಸಂಖ್ಯೆಯನ್ನು ಹೆಚ್ಚು ಮಾಡಬೇಕಾದ್ರೆ, ಅಥವಾ ತುಂಬಾ ಹೆಚ್ಚಾಗಿದೆ ಅಂತಂದ್ರೆ ನಾವು ಬೇರೆ ಮತ್ತೆ ವಿಸ್ತರಣೆ ಮಾಡಲಿಕ್ಕೆ ಸಾಧ್ಯ ಉಂಟಾ, ಅಥವಾ ಒಂದು ಪ್ರದೇಶದಲ್ಲಿ ನಿರ್ದಿಷ್ಟವಾಗಿ ಎಷ್ಟು ಪ್ರಾಣಿಗಳನ್ನು ನಾವು ಹೊಂದಬಹುದು, ವಾಟ್ ಆರ್ ದಿ ಕ್ಯಾರಿಂಗ್ ಕೆಪಾಸಿಟಿ ಆಫ್ ಎ ಪರ್ಟಿಕ್ಯುಲರ್ ಟೈಪ್ ಆಫ್ ಫಾರೆಸ್ಟ್ ಫಾರ್ ಎ ಪರ್ಟಿಕ್ಯುಲರ್ ಸ್ಪೀಶೀಸ್ ಆಫ್ ಎನಿಮಲ್ಸ್. ಈ ತರ ಪ್ರಶ್ನೆಗಳಿಗೆಲ್ಲ ಉತ್ತರ ಕೊಡಲಿಕ್ಕೆ it is very important to accurately determine, accurately estimate wildlife populations. ಅದಲ್ಲದೆ ನಾವು ಈ ಪರಿಸರ ಮತ್ತೆ ನೇಚರ್ನ ಅರ್ಥ ಮಾಡಿಕೊಳ್ಳಲಿಕ್ಕೋಸ್ಕರ ಕೂಡ, ಅಂದ್ರೆ ವೈಜ್ಞಾನಿಕವಾಗಿ ಅರ್ಥ ಮಾಡಿಕೊಳ್ಳಲಿಕ್ಕೆ ಕೂಡ ಎಷ್ಟಿದೆ, ಎಷ್ಟರ ಮಟ್ಟಿಗೆ ಬೇರೆ ಬೇರೆ ಹ್ಯಾಬಿಟ್ಯಾಟ್ ಟೈಪ್ನಲ್ಲಿ ಒಂದೇ ಜಾತಿ ಪ್ರಾಣಿ ಯಾವ ರೀತಿಯ ಸಾಂದ್ರತೆಗಳು ಹೊಂದಲಿಕ್ಕೆ ಸಾಧ್ಯ, ಇಂಥದನ್ನು ಕೂಡ ನಾವು ಒಂದು ವೈಜ್ಞಾನಿಕ ತಳಹದಿಯ ಮೇಲೆ ನಾವು ನಿರ್ಧಾರ ಮಾಡಬೇಕಂದ್ರೂ ಕೂಡ it becomes very important to estimate animal populations reliably. ಸೊ ಇದನ್ನ ಮಾಡಬೇಕಾದ್ರೆ ಬಹಳ ಇಂಪಾರ್ಟೆಂಟ್ ಅಂದ್ರೆ ಆ ಗಣತಿಕಾರರ ನಡುವೆ there should be high levels of motivation, there should be data integrity, otherwise what numbers you get will not be meaningful. ಅದಲ್ಲದೆ ಪ್ರತಿಯೊಂದು ವಿಧಿ ವಿಧಾನಗಳಿಗೆ ಅದಕ್ಕೆ ತಕ್ಕಾದ ಒಂದು ರಿಸೋರ್ಸಸ್ಗಳು ಬೇಕಾಗುತ್ತೆ, ಟ್ರೈನಿಂಗ್ ಆಗಿರೋ ಅಬ್ಸರ್ವರ್ಸ್ ಬೇಕಾಗುತ್ತೆ, ಅಥವಾ ಯು ನೀಡ್ ಪೀಪಲ್ ಹೂ ಆರ್ ಟ್ರೈನ್ಡ್ ಇನ್ ದಿ ಅಕಾಡೆಮಿಕ್ ಮೆಥಡ್ಸ್ ಆಸ್ ವೆಲ್ ಆಸ್ ದಿ ಫೀಲ್ಡ್ ಸ್ಕಿಲ್ಸ್. ಅದನ್ನೆಲ್ಲ ನೋಡಿಕೊಂಡು ಈ ಒಂದು ಜಾಗಕ್ಕೆ ಯಾವ ವಿಧಿವಿಧಾನ ಅಪ್ಲೈ ಆಗುತ್ತೆ ಅನ್ನೋದು ಕೂಡ ಇಂಪಾರ್ಟೆಂಟ್. ಮತ್ತೊಂದು ಬಹಳ ಇಂಪಾರ್ಟೆಂಟ್ ಏನು ಅಂತಂದ್ರೆ, ವನ್ಯಜೀವಿ ಗಣತಿ ಅಂದ ತಕ್ಷಣ ಸಾಮಾನ್ಯವಾಗಿ ಎಲ್ಲರನ್ನು ಕೂಡ ಪೂರ್ಣ ಗಣತಿ, ಸೆನ್ಸಸ್ ಅಂತ ಯೋಚನೆ ಮಾಡ್ತಾರೆ. ಅದು ತಪ್ಪು. ಈಗ ಇಫ್ ಯು ವಾಂಟ್ ಟು ಡಿಟರ್ಮಿನ್ ಹ್ಯೂಮನ್ ಪಾಪ್ಯುಲೇಷನ್, ದೆನ್ ಸೆನ್ಸಸ್ is okay, because you are going to visit every household, you will go, you will make a census, and you will, through questionnaire surveys, you will collect data, which means 100% of the area you cover, all the households are visited and the numbers are collected. Whereas sampling ಅಂತಂದ್ರೆ ಪ್ರತಿಚಯನ ಪದ್ಧತಿ, wherein you adopt a sampling technique so that you actually survey a part of an area which is representative of the whole habitat or a whole region, and based on that information, based on the data that you collect, you try to extrapolate it to the entire area of interest. So census is a complete count, whereas sampling is different from census. So we will see, how sampling is actually more relevant when it comes to estimating wildlife populations. ಇದರಲ್ಲಿ ಎರಡು ಬಹಳ ಪ್ರಮುಖವಾದ ವಿಚಾರಗಳಿದೆ. ಒಂದು, when you actually go and visit a forest, because of the vegetation, you will not be able to see a lot of you will not be able to see all animals. You will only see a part of them. So there is some animals which are actually missed by the people who are estimating, either because of the vegetation or because of other problems. So it is very important to recognize this fact that we can't see all animals out there in the forest. We can only see a fraction. What is that fraction? So we need to estimate that fraction, or the fraction which we are unable to see. That is called the observability or detectability. The second important point, is spatial sampling. Basically, it means you can't be there everywhere. ಒಂದು ವಿಶಾಲವಾದ, ಈಗ ಒಂದು ನಾಗರಹೊಳೆ ಅಂತ ನಾವು ತಗೊಂಡ್ರೆ ಅದು 640 ಸ್ಕ್ವೇರ್ ಕಿಲೋಮೀಟರ್ ಇರುವಂತಹ ಒಂದು ವಿಶಾಲವಾದ ಪ್ರದೇಶ. You will not be able to go and visit the entire forest. You will only be able to assess or estimate numbers at a fraction of an area out there. So this is again we have to recognize. So we are only estimating the numbers from a fraction of an area, and then using statistical methods, we will extrapolate it to the entire area of interest. So any method that ignores these two fundamental problems, you are sure that that method is not going to be reliable. ಅದು ಖಂಡಿತ ವಿಶ್ವಾಸಾರ್ಹವಾದ ಪದ್ಧತಿ ಅಥವಾ ವಿಧಿವಿಧಾನ ಆಗಲ್ಲ. ಸೊ ಯಾವುದೇ ಒಂದು ವನ್ಯಜೀವಿ ಗಣತಿಯಲ್ಲಿ this partial observability, and we are only sampling a part of the space or a part of the habitat. So these two become very, very important factors in wildlife population estimation. So just to give you an example here, if whatever animals that are out there, ಇದು ಕೆಂದಳಿಲು ಅಂತ ಹೇಳ್ತೀವಿ, ಮಲಬಾರ್ ಜೈಂಟ್ ಸ್ಕ್ವಿರಲ್, just to give you an example. Let's say, you are walking in the forest on this line. Let's say there are some animals which you will be able to see, and there are some other animals which you are unable to see. So these individuals, you are missing them, whereas these individuals, you are able to count them. So it becomes very important to actually use a method that, using the information that you have obtained from the sightings of these animals, you should be able to estimate what fraction of the animals that we are missing. This is the biggest challenge in wildlife population estimation. So just to put it, in a very simple equation, if n is the number of animals that we want to estimate in a given area, C becomes a count statistics. That is the number of animals that you are able to count. And alpha is the proportion of the area of interest that you are able to actually survey. Then P is the estimated detection probability. That is the partial observability ಅಂತ ನಾನು ಹೇಳಿದ್ನಲ್ಲ. So that is the one. So if you have count statistics, that is the number of animals counted in a given survey, then if you know the proportion of the area surveyed, and if you are able to estimate the detection probability, that is the probability of seeing animals, then you will be able to reliably estimate the number of animals in a given area. This is actually called the canonical estimator. ಇದಕ್ಕೆ ಬೀಜ ಮಂತ್ರ ಅಥವಾ ಮೂಲ ಮಂತ್ರ ಅಂತ ಹೇಳ್ತಾರೆ. So ವನ್ಯಜೀವಿ ಗಣತಿಗೆ this becomes really important. Any method that deals with this, there may be so many methods, for different species or different taxa, but this is the most fundamental one, that as long as the method accommodates this challenge, then your estimation method will be very reliable. So let's just take an example. Let's say that we adopt some method and we are able to count the number of sambar deer in a area. Okay, so there are 30 sambars that you are able to count. So C is 30, and you are able to actually sample only 40% of a habitat, let's assume. So the sampling, so the spatial sampling, what we mentioned, that is the alpha, that is the fraction of the area that we are sampled, is 0.4. And let's also, let's say, that through a very good method, we are able to estimate the detection probability 0.5, which means we are able to see only 50% of the animals that are present there. So the detection probability is 0.5. The fraction of the area sampled is 0.4, and the count is 30. So this becomes very straightforward. So which means even though you have counted only 30 sambar deers, because you know detection probability, you know the sampling fraction, you are able to estimate that there are 150 sambar deers are there in this area. I hope it is pretty clear to you all now. Okay, so this is a very simple example. So this is called Mula Mantra athwa canonical estimator of abundance when it comes to estimate abundance of wildlife populations. Now the challenges are there, as I told you, that all that can be counted cannot be seen. You know that there are deers out there, but you cannot see, you can only see a part. You cannot be there everywhere to count everything. There are many methods to count, but there are only a few methods to estimate animal numbers. Some of you may have heard of block counts, drive counts, water hole counts. ಅಂದ್ರೆ ಎಲ್ಲಿ ಎಲ್ಲಿ ವಾಟರ್ ಹೋಲ್ಗಳು ಇರುತ್ತೋ, ನೀರಿರೋ ಜಾಗದಲ್ಲಿ ಅಲ್ಲಿ ಒಂದು ಕೂತ್ಕೊಂಡು ಅಲ್ಲೆಲ್ಲ ಗಣತಿ ಮಾಡ್ತಾರೆ. ಬಟ್ ಅಲ್ಲಿ ಎಲ್ಲಿ ವಾಟರ್ ಹೋಲ್ ಇಲ್ವೋ ಅಲ್ಲಿ ಎಷ್ಟಿದೆ ಪ್ರಾಣಿಗಳು ಅನ್ನೋದು ನಿಮ್ಗೆ ಗೊತ್ತಾಗಲ್ಲ. ಸೊ ಈ ಮೆಥಡ್ನಲ್ಲಿ ಈ ಬ್ಲಾಕ್ ಕೌಂಟ್ಸ್ ಆಗಲಿ, ಡ್ರೈವ್ ಕೌಂಟ್ಸ್ ಆಗಲಿ, ವಾಟರ್ ಹೋಲ್ ಕೌಂಟ್ಸ್ ಆಗಲಿ, ದೇ ಆರ್ ನಾಟ್ ಸ್ಯಾಂಪ್ಲಿಂಗ್ ಬೇಸ್ಡ್ ಮೆಥಡ್ಸ್. So whatever numbers that you derive through this method will not be very reliable. And of course, that ultimately whatever animals that we are interested in, we should know the biology of that animal so that we can adopt the correct method. So typically an estimation of wildlife population involves, we have to design a survey based on some sampling scheme. It can be random sampling, stratified sampling, so many different sampling methods are there. Then there has to be a survey protocol from which you are able to estimate the detection probability. Then we have to go and do the survey and count animals, and perhaps also make the other associated measurements or observations which will help you to actually estimate P and alpha that I talked to you about. Then we have to adopt a statistical method, and we have to choose a good model that fits the data. Then also, when you are estimating any parameter, you should always estimate also the associated precision or bias that are there. This is part of the statistics. So some of the most important parameters that you use is the central tendency, or it is called mean or average. You have heard of it. You might have also heard about standard deviation or standard error. ಅಂದ್ರೆ ಇದು ಎಷ್ಟರ ಮಟ್ಟಿಗೆ ವೇರಿಯಬಲ್ ಆಗಿದೆ, ನಾವು ಯಾವುದೇ ಒಂದು ಎಸ್ಟಿಮೇಟ್ ಮಾಡಿದಾಗ ಒಂದು ಸರಾಸರಿ ಎಷ್ಟು ಮತ್ತೆ ಅದು ಎಷ್ಟರ ಮಟ್ಟಿಗೆ ವೇರಿಯೇಷನ್ ಆಗುತ್ತೆ ಅನ್ನೋದು ಈ ಎರಡು ಸ್ಟ್ಯಾಟಿಸ್ಟಿಕಲ್ ಪ್ಯಾರಾಮೀಟರ್ಸ್ ಇನ್ ಮೆಷರ್ಸ್ ಇಂದ ನಮಗೆ ಗೊತ್ತಾಗುತ್ತೆ. And with that, then ultimately you will be able to make useful conservation decisions. Okay, so now let's quickly go through two major methods. One is the distance sampling method, another one is the capture recapture sampling method. What are they? So I will again quickly refer you back to n is equal to c divided by p alpha. ಸೊ ಇದರಲ್ಲಿ ಕ್ಯಾಪ್ಚರ್ ರಿಕ್ಯಾಪ್ಚರ್ ಸ್ಯಾಂಪ್ಲಿಂಗು ನಲ್ಲಿ ಏನು ಹೇಳುತ್ತೆ ಅಂದರೆ, you are actually going to put a number of camera traps, let's say for the example of a tiger, in some sampling way, so that you are able to obtain the number of individual animals that are obtained in your camera traps. So here P refers to the capture probability. That is the probability of capturing an individual animal. So the method should be able to estimate this. How do we do this? Okay, so let's get into a bit more detail. So capture recapture sampling is used for animals that we cannot see or measure distances. ಅಂದ್ರೆ ನಾನು ಹೇಳಿದ್ನಲ್ಲ, ಈಗ ಮಾಂಸಾಹಾರಿ ಪ್ರಾಣಿಗಳು ಮತ್ತೆ ಸಸ್ಯಾಹಾರಿ ಪ್ರಾಣಿಗಳು ಅಂತ. ಸಸ್ಯಾಹಾರಿ ಪ್ರಾಣಿಗಳು ನಿಮಗೆ ನೋಡಲಿಕ್ಕೆ ಸಲೀಸಾಗಿ ಸಿಗುತ್ತೆ, ಅದನ್ನು ನೀವು ನೋಡಿ ಅದರ ಬಗ್ಗೆ ಏನಾದರೂ ಅಬ್ಸರ್ವೇಷನ್ಸ್ ಮಾಡ್ಬೋದು. ವೆರಾಸ್ ನಾಕ್ಟರ್ನಲ್ ಸೀಕ್ರೆಟಿವ್ ಎನಿಮಲ್ಸ್ ಯು ಆರ್ ನಾಟ್ ಏಬಲ್ ಟು ಸಿ ಅಟ್ ಆಲ್. ಅಂಥ ಪ್ರಾಣಿಗಳಿಗೆ ಈ ಕ್ಯಾಪ್ಚರ್ ರೀಕ್ಯಾಪ್ಚರ್ ಸರ್ವೆ ಟೆಕ್ನಿಕ್ನ ಉಪಯೋಗಿಸಲಾಗುತ್ತೆ. ಸೊ ಇದರಲ್ಲಿ ಕ್ಯಾಚ್ ಅಂತಂದ್ರೆ ಅರ್ಥ ಏನು ಅಂದ್ರೆ, ಹೇಗಾದರೂ ಮಾಡಿ ನಾವು ಆ ಪ್ರಾಣಿಗಳನ್ನ ಹಿಡಿಬೇಕು. ವೆನ್ ಯು ಸೇ ಕ್ಯಾಚ್, it doesn't necessarily mean catching physically. If it is possible, it is different. For example, fishes, let's say you are interested in estimating the number of fishes in a pond. Let's say you are able to catch fish, then you mark them, you release them back, then you again catch them, and next time when you catch them you see how many marked fishes were there. Then with that frequency, with that number, you will be able to estimate the number. Let me tell you a little bit more detail about this. ಅಂದರೆ ಪ್ರತಿಯೊಂದು ನೀವು ಮಾಡಿದಾಗ ಒಂದು ಗುರುತು ಹಾಕೋದು ಇಂಪಾರ್ಟೆಂಟ್. ಆ ಗುರುತು ಹೇಗೆ ಹಾಕ್ಲಿಕ್ಕೆ ಸಾಧ್ಯ? ಬರ್ಡ್ಸ್ಗಾದ್ರೆ ಟ್ಯಾಗ್ ಹಾಕ್ತಾರೆ, ರೋಡೆಂಟ್ಸ್ಗಾದ್ರೆ ಅದು ಕಿವಿನಲ್ಲಿ ಒಂದು ಗುರುತು ಮಾಡ್ಬೋದು, ಅಥವಾ ಕೆಲವು ಪ್ರಾಣಿಗಳಿಗೆ ನ್ಯಾಚುರಲ್ ಮಾರ್ಕಿಂಗ್ಸ್ ಇರುತ್ತೆ. ಫಾರ್ ಎಕ್ಸಾಂಪಲ್, ಹುಲಿಗಳಿಗೆ ಮೈ ಮೇಲೆ ಪಟ್ಟೆ ಇರುತ್ತೆ. ಆ ಪಟ್ಟೆ ಪ್ರತಿಯೊಂದು ಹುಲಿಗೂ ಕೂಡ ಬಹಳ ವಿಶಿಷ್ಟವಾಗಿರುತ್ತೆ. There are no two individual tigers in the wild which have the same stripe pattern. Even mother and the cub also will not have the same pattern. So it is just like your fingerprints. There are no two people on earth who have the same set of fingerprints. Every fingerprint is unique. It is like that. So the natural markings that you see on tigers, or the rosette patterns that you see on leopards, are unique to that individual. So if you are able to take a picture of a tiger or a leopard through some means, and if you are able to identify them based on that, then it becomes very easy. Now the technology has also improved. You are able to actually extract DNA from blood or hair or the droppings of an animal. Then you will be able to accurately identify the individual. So there are so many different methods, wherein you can accurately identify individual animal. That becomes a very important aspect of this method. ಅಂದ್ರೆ ಈ ಮೆಥಡ್ ನಲ್ಲಿ ನೀವು ಅದು ವರ್ಕ್ ಆಗಬೇಕು ಸರಿಯಾಗಿ ಅಂದ್ರೆ ಪ್ರತಿಯೊಂದು ಪ್ರಾಣಿ ಇಂಡಿವಿಜುವಲ್ ಆಗಿ ಅದನ್ನ ಗುರುತು ಹಿಡಿಲಿಕ್ಕೆ ಆಗಬೇಕು. ಈಗ ನಾವು ಎರಡು ಪಿಕ್ಚರ್ ತಗೊಂಡಿದ್ದೀವಿ ಅಂತ ಇಟ್ಕೊಳ್ಳಿ, ಹುಲಿದು. ಇದನ್ನ ನೀವು ಕಂಪೇರ್ ಮಾಡಿ ನೋಡಿ, ಯಾವ ಬೇಕಂದ್ರೆ ಈವನ್ ಎ ಪ್ರೈಮರಿ ಸ್ಟೂಡೆಂಟ್ ವಿಲ್ ಬಿ ಏಬಲ್ ಟು ಕಂಪೇರ್ ಅಂಡ್ ಯು ಕ್ಯಾನ್ ಸಿ ಈ ಗುರುತು ಹೇಗಿದೆ, ಈ ಗುರುತು ಹೇಗಿದೆ, ಈ ಪಟ್ಟೆ ಗುರುತು ಹೇಗಿದೆ, ಈ ಪಟ್ಟಿ ಗುರುತು ಹೇಗಿದೆ, ಇದು ಅದೇಯಾ ಇದು ಬೇರೆಯಾ. ಸೊ ಯು ವಿಲ್ ಬಿ ಏಬಲ್ ಟು identify very clearly, say that these two are different individuals. So it becomes very important to actually identify the individual animals accurately. If that is possible, then it is possible to estimate the number. Let's say n is the number of individuals in an area which you don't know, which you want to estimate. P is the probability of the capture. Let's say, first time you go and make an attempt, you get some number of animals. You are able to catch some number of animals and you are able to mark them, identify them, let's say. That is N1. And second time, let's say, you are marked them and you have released them. And second time again, you try to catch them. Then you get a number of animals that you are able to catch during the second sample, that is N2. Then M becomes the number of animals which are common between the two samples. So if you have this information, then you will be able to estimate the number of animals in a given area. It is given through a very simple formula, n1 into n2 divided by m. ಇದರ ಒಂದು ಎಕ್ಸಾಂಪಲ್ ಕೊಡುವ ಅಂತಂದ್ರೆ, let's say that that you are able to catch 10 fishes. You are able to mark them and you are able to release them back. Next time again, let's say you are able to catch 12. ಎರಡನೇ ಸಲ ನೀವು ಪ್ರಾಣಿಗಳನ್ನ ಹಿಡಿದಾಗ ಅಥವಾ ಈ ಮೀನುಗಳನ್ನ ಹಿಡಿದಿದ್ದೀರಾ ಅನ್ಕೊಳ್ಳಿ, ಅದರಲ್ಲಿ 6 ಮೀನುಗಳು ಮಾತ್ರ ಮುಂಚೆ ಮಾಡಿದ್ದ ಮಾರ್ಕ್ ಇತ್ತು. ಅಂದ್ರೆ 6 ಹೊಸ ಮೀನುಗಳು ಮತ್ತೆ 6 ಮುಂಚೆಯೇ ಮಾರ್ಕ್ ಮಾಡಿದ್ದಂತ animal. ಅಂದ್ರೆ ದಿ number of individuals which are common between n1 and n2 is 6. So n will become 10 into 12 divided by m. ಎಷ್ಟಾಯ್ತು? 20 ಪ್ರಾಣಿಗಳಾಯ್ತು. So this is called the basic estimator or the Lincoln-Peterson estimator. This is the most fundamental equation that is actually used when you are actually having only 2 samples. Then there will be certain important assumptions that it has to be done. For example, it should be done in a short period of time so that there are no additions to the populations or there are no deletions to the populations. And we also need to, every individual animal should have some probability of capturing them. And also the other important thing is that we should not lose the mark. For example, if you are not able to actually clearly anamnetically identify the animal, then you will, let's say once you have marked the animal and you have released it, next time when you get it, even though the mark was there, but let's say you are not able to find the mark, or the mark has got erased, or the tag has got dropped, then this method will not work. So what I will do is, instead of actually getting into more technical details, what I would like to say, is that in a given area, let's say if you are interested in tigers, so what we do, is that we survey the entire area for looking for roads and trails where you expect tigers to move around. Then we put a number of camera traps. These are all remotely sensed camera traps. ನೀವು ಅಲ್ಲಿ ಕೂತ್ಕೊಂಡು ಫೋಟೋ ತೆಗೆಯೋದಲ್ಲ, it is basically a remotely operated camera traps. These camera traps are placed in a number of locations. When the tiger actually moves on that road, it will be sensed by the camera either through an IR beam or through a motion sensor. Then it automatically triggers the camera and takes the picture. You typically put two cameras on either side, so that when the tiger goes, you get both left flank and the right flank pictures, because the stripe patterns are different for the same individual. Both right side stripe pattern is different from the left side. That is why if you want to accurately identify the individual, it becomes very important to have two cameras on either side of the trail or a road, so that you simultaneously get two pictures, right picture and the left flank, so that you can accurately identify them. Then you use the statistical method to estimate the number of animals. Okay, so that is how these are actually done in the field. Number of camera traps are deployed, so many pictures are obtained, you compare them. Earlier, we used to compare them manually, but now there is a software which compares the stripe patterns and says whether it is the same or different. So you will be able to accurately estimate how many individuals that you have got in the camera trap. Then using a statistical method, you will be able to identify how many are common between the samples, and looking at the frequency, you can estimate n. I have tried to simplify the entire method to you just to get you an idea of what is camera trapping, how do you actually estimate the number of tigers. Okay, and once you are able to do it over time, you can say whether the population is increasing or whether the population is decreasing or whether it has remained the same over the years. The second method is the distance sampling, or this is used for herbivores, ಸಸ್ಯಾಹಾರಿ ಪ್ರಾಣಿಗಳಿಗೆ ಉಪಯೋಗಿಸುವಂತದ್ದು. ಇದರಲ್ಲಿ this is again, if you remember, this is the ಮೂಲ ಮಂತ್ರ ಅಂತ ಹೇಳಿದ್ನಲ್ಲ, canonical estimator. But here now C refers to the number of animals that you are able to count when you are actually walking on a transact line. Then P becomes the detection probability, and alpha again is the sampling fraction. Let us again see what assumptions are needed. So this distance sampling is slightly different from the capture recapture sampling. Either see, capture recapture was applicable for those which are nocturnal, secretive, you are not able to see. Whereas for animals like herbivore animals, large mammals which you can readily see, you can actually see them in the forest. When you walk in the forest, you will be able to count those animals. But you still don't get all the animals, but you get only a portion of the animals or a fraction of the animals out there. So the main assumptions here are there, that whatever the transact lines or the trails that you are able to establish in the forest for walking, that should be random lines. And then any animal which is there on the line or close to the line are detected with certainty, which means the probability of detecting an animal if it is on the line is 1, which means you are very certain that you will be able to detect them. And as the animal is away from the line, their detection probability decreases. And then whenever animals are there, before they run off, you should be able to take the measurements. So it is important to take certain measurements. I will tell you what those measurements are at the initial locations. And these measurements typically are called sighting distances and sighting angles. What are they? Let me explain that to you. So let's say, if this is a transact line, if you are able to see all animals, then it is easy. You can just count the number of animals that you are able to see. You are able to see everything. You know the length of the walk. You know the width within which you are able to see animals. So it is simply, it is a rectangular area you have sampled, length into width, and the number of animals that you have counted. So you know what is the number of animals per unit area. But the thing is, we are talking about a situation in forest where you see only a few, you are not able to see some others. So these are all individuals which are missed. These are individuals which you have been able to see and count them. So in this method, what we do is we count the number of animals that you are able to see. You know what is the distance that you have traveled. Then, based on the distance, based on the animals that you have detected from this line, we estimate what is the perpendicular distance between the animal sighted and the line. If you have that information, then I can take an average perpendicular distance within which I am seeing the animals. So you are able to get the average width of the area that you are sampling. Therefore, you will be able to estimate the number of animals in a much more reliable way. So here, n becomes the count, that is the number of animals you are able to count. P is the detection probability. W is the width, half width. We are seeing on both sides of the line, so it becomes 2 into W, and L is the total length that you have walked. So this, is the basic formula for calculating animal abundance using distance sampling method. But how do you actually estimate the perpendicular distance? Let me give an example. Two people are walking in the forest along a predetermined line, which is called the transect line. You see a group of animals, you count the number of animals in the group, you make a note, you record them. Then you also actually measure using a range finder, which measures the distance from you to the animal. This is called the sighting distance. Okay, so there are both optical rangefinders and laser rangefinders. You will be able to accurately estimate or accurately measure the distance from you to the group center of the animal. Then, using a compass bearing, you also make a note of the bearing in which you are going, that is the degree, okay, with respect to north. So once you get, let's say, you are walking in east direction, so that is 90 degrees east. Let's say that the angle with respect to the compass bearing where you have seen, let's say it is 45 degrees. So this is 90 degrees, this is 45 degrees. So this is the sight angle. This minus this is the sighting angle. So if you know the diagonal distance, if you know the angle, then using the Pythagoras theorem, you can actually calculate what is the perpendicular distance between the group center of the animal and the line. Okay, so here it will be perpendicular distance will be equal to r sine theta. This is the distance and sine theta. Okay, so you will be able to estimate very clearly what is the perpendicular distance. And once you have perpendicular distances to all the animals, that whatever you were able to see, you are not able to measure for those which you have not seen them, that is fine. But whatever group of animals that you have seen, if you are able to measure the perpendicular distance through this field method, then you get a number of perpendicular distances, and you can take an average perpendicular distance within which you are seeing the animals. So that will give you the effective strip width, and then you have the total distance walked. Then you will be able to estimate the number of animals in a given area. So this is the basic concept. Then we actually use a statistical method and a sampling method to estimate the number of deers, number of pigs, or number of elephants in a given area. So this method, was actually pioneered, by Dr. Ulas Karant in way back in 86, 87. This line transact technique was implemented in Nagrole. Whereas in 1990, capture recapture technique for the first time anywhere in the world, it was actually adopted by Dr. Ulas Karant in Nagarhole. With that, now these two methods have become very common. Now it is applied in so many places, not only in India but across the world. So you get a very good accurate estimate of the number of animals in this method. This is just to show you, in the different sites, Bandipur, Nagarhole, Badra, BRT, how transacts are marked and they are all walked, like in each year we thousands of kilometers, are walked to collect information about the number of animals that you are able to count. Then using the method, you can actually estimate the numbers and then say again whether they are increasing or decreasing. So this is the distance sampling method. So now I thought, that uh, I will I will just show you a video of how it is actually done, in the forest through a video. Uh, let's see whether we are able to do that. You are able to hear them, I hope.
Yes, sir, we can hear. Thank you, sir.
The forests of Asia are home to one of the most endangered mammals on Earth, the tiger. Every year, large amounts of money and manpower are invested for the conservation of the species. But how can we tell if these investments are working? Only through monitoring if tiger populations are going up, going down, or holding steady. This training video will demonstrate modern, scientific methods for monitoring tigers and their prey. Monitoring tigers is by no means easy, for they are secretive predators that are rarely seen, except in a few reserves where some tigers have become habituated to tourism. A majority of tigers lead solitary, hidden lives in thick jungle and are often nocturnal in their habits. How then do we keep track of their numbers? Since tigers don't live in communities, we can't simply go from cave to cave and conduct a census. Fortunately, there are other ways, and biologists like Dr. Ullas Karant of the Wildlife Conservation Society have developed reliable, scientific methods for estimating tiger numbers. If these methods are followed meticulously, it is possible to keep track of tiger populations. However, even the best methods should not be practiced blindly. To produce reliable results, they have to be combined with a basic knowledge of tiger biology, such as how much space do tigers need? What kind of habitat do they prefer? What do they eat and how much? One important fact is that there is a direct correlation between the number of prey animals in a forest and the number of tigers the forest can support. Each adult tiger needs about 3,000 kilograms of live prey every year. They need large-sized prey and lots of it. To survive, a tiger needs to kill about 50 to 60 deer-sized prey animals per year. So, for every single tiger in a forest, there needs to be a population of at least 500 prey animals that produce a surplus of 50 animals for the tiger to eat each year. Therefore, counting the number of prey animals in a forest will give us a rough idea of how many tigers the forest can support. That's why Monitoring tiger numbers also involves counting the number of prey animals in a forest. But this is easier said than done, as prey animals are alert and elusive. Often they live in thick jungle and are not easily seen. Therefore, animal censuses through direct counts do not work in forested landscapes. We can only estimate animal numbers, and to do this reliably, we need to use well-tested sampling techniques. Here is how the sampling method works for estimating prey animals in a forest. If we take a sample area of forest and thoroughly count the number of prey animals in it, we can extrapolate this information to estimate the number of prey animals in the entire forest. However, this is complicated by the fact that forests often have different types of habitats with differing concentrations of herbivores. For instance, one part of a forest may have a high density of prey animals, and another part could have a low density of prey animals. If we only sample the low-density area and extrapolate this information to the entire forest, then we will end up underestimating the number of animals. On the other hand, if we sample only the high-density area and extrapolate that information to the entire forest, we will end up with an overestimate. So, to get a reliable overall estimate, it is necessary to sample from all the representative habitats. There is one more factor to keep in mind. Even within the area that we sample, it is impossible to count all the animals that are present. Because in a forest, some of the animals are seen and some remain hidden. So only a certain proportion of the total number of animals present can be detected by us, while others go unseen. This is why we need to use sampling techniques. The scientific technique described in the next chapter is one of the reliable methods for sampling prey species. One of the best methods of sampling prey species is through a technique known as line transect sampling. A line transect is a straight line through a forest on which observers walk to count animals on both sides of the line. The technique involves not only counting the numbers of animals seen, but also measuring their perpendicular distance from the transect line. Transects can be simple straight lines that go from point A to point B, or they can take the shape of squares or triangles. The advantage of a triangle or a square is that the field workers come back to the same point where they started. This makes logistics easy, as people have to be dropped and picked up at transects. When designing line transect surveys, it is important to ensure that transects are situated at random with relation to the distribution of animals. If transects are deliberately situated along waterholes or salt licks, we will end up with an overestimate. For more information on this, please refer to chapters 9 and 10 of the manual. Line transect sampling can be accomplished using volunteers and wildlife staff, but only after they have been trained properly. Field workers should be trained in the concept and practice of sampling by an expert. In addition to theoretical training, a short stint of field training too is necessary with an experienced instructor. Apart from learning the correct techniques, it is imperative that field workers are able to correctly identify the prey species they encounter. In Asian forests, these might be gaur, sambar, spotted deer, wild pig, and muntjac. As we saw earlier, because of trees, bushes, and shrubs in a forest, some animals will be seen and others will go unseen. This is not a problem for estimation. However, it is vital that the animals that are seen are counted carefully, and the distance from the observer to the animals is measured accurately. In line transect sampling, all animals that are on the line or immediately adjacent to it are detected. But as the distance increases from the transect line, the detectability of animals gets reduced due to vegetation or because animals often blend in with the background. Factors like these are taken into consideration in the scientific calculations that will be used later. Before observers can walk on a transect line, the line must first be marked through the habitat. The line must be straight and can be indicated with the use of red paint on trees. It is important that the observers are able to see the line at all times. The vegetation must not be disturbed too much or cut unnecessarily, as that may alter the results of the sample. It is sufficient if there is just enough space for the observers to walk silently in single file. Walking on a transect line needs care. As far as possible, the observers must see the animals before the animals see them. If the animals scatter before they can be detected, it is of no use. So, the observers must wear camouflaged clothes and walk silently through the habitat. Proper footwear is essential for walking in the jungle. Extra protection for the legs can help prevent ticks or leeches from getting on. The trousers on shirt or jacket must help the observer blend with the surroundings. A few items of equipment are also required. Binoculars for identifying animals clearly and making accurate counts. A rangefinder to measure the distance from the observer to the animals. A compass. And of course, one needs a data sheet to note down the observations. Walking on a transect needs care and practice. The team should consist of only 2 people. They should walk silently and at a uniform pace of 1.5 to 2 kilometers an hour. They should remain alert for observing prey animals. The person walking in front scans the area right ahead. The person walking behind scans the area not covered by the first person. When any animals are spotted, the observers quickly identify and count the animals and measure the distance to the animals using the rangefinder. It is crucial to measure the exact distance of the animals from the transect line. Any mistakes made will alter the results of the estimate. Since we usually see animals well before we come abreast of them, measuring perpendicular distance from the transect line needs a special technique. That's why one observer also carries a compass. After measuring the observer to animal distance, a compass reading of the animal's position in relation to the transect line should be taken. The measured distance and the angle will enable us to calculate the perpendicular distance later. Please note that when there is a group of animals present, the measurement should always be made to the center of the group or herd. Please also note that if the animals have moved since they were first spotted, the measurement should always be taken to the point where the animals were first spotted and not to the new position. All data, such as number of animals for each sighting, distance from the observer, and the compass bearing must be noted down meticulously in the data sheet before the observers move on. The observers must not linger watching the animals, but must move on as soon as they have collected these data. The best time to walk on a transect line is during early morning and late afternoon hours when prey animals are most active. The best time of the year for transect surveys is during the dry season, when the forest is most open and visibility is good. It is even better after a few rain showers so that the leaf litter does not make too much noise while walking. A transect is usually 3 to 4 kilometers in length. In some cases, a transect may have to be walked repeatedly to get proper estimates. It may not be enough to walk on a transect line just once. It is extremely important to have as many transects as possible to sample the entire habitat. An ideal number would be between 20 to 40 transects. All data that are collected in the field are analyzed in a computer software program known as distance. This software takes in raw data and generates prey animal density estimates. In summary, the following points must be remembered. Line transect sampling helps us estimate the number of prey animals in a large forest area. The objectives in a line transect survey are to identify prey species, count the number of animals encountered at each sighting, and measure their distance from the observer standing on the transect line. Since animals are often seen before we come abreast of them, two measurements must be taken with every sighting: the distance from the animals to the observers and the angle. These two measurements will later enable us to calculate the perpendicular distance. Whenever there is a group of animals, the distance measurement must always be made to the center of the group. If the animals have moved from the time they were first spotted, the measurement should be made to the place where they were first spotted and not to the new position. All measurements must be accurate and they must be noted down correctly in the data sheet. Once the counting and measuring is done, the observers must immediately carry on. While on a transect, silence should be maintained. Smoking is strictly prohibited. The observers must walk at an even pace of 1.5 to 2 kilometers per hour. Only animals that are seen and correctly identified must be counted. Since there are dangerous animals in Asian forests, field workers must take appropriate common sense precautions while walking on transect lines. For more information, please refer to chapters 9 and 10 of the manual Monitoring Tigers and Their Prey by K. Ullas Karant and James D. Nichols.
Okay, I think we are just running out of time, so this gives a good idea about what is transact, what is distance sampling, how do you really estimate the number of animals, particularly herbivore animals in the forest. So whenever there is another opportunity, perhaps we can talk more about how the tigers are estimated using capture recapture sampling. Hope you found this talk useful. If there are any questions, I'll be happy to answer.
Thank you very much,
Umesh?
sir.
Yeah.
Yes, sir. There is one question. You can take that. Yes, sir.
Okay.
Question from Dr. Netravati. Is it true that the species richness decreases from superficial forest area to the core forest area?
What decreases?
Species richness.
Oh, species richness.
Yes.
Okay. Okay. See, one thing I want to clarify is that we are talking about the number of animals, that is the abundance or population density in this talk. Species richness refers to the number of species in a given area. So, the concept of species richness is different from population density or abundance. Now, having said that, the question is that whether the species richness changes in superficial area versus core area. First of all, this core, buffer, all these, are basically management units, are these are all the definitions made by us. So what it actually means is that when you say core, it practically means that they are undisturbed original forests. And when you say superficial, it is heavily human modified forest. If you actually mean that way, then it is definitely true in a sense that the species richness between undisturbed forest and heavily human disturbed forest will be different. And it again depends on what taxa you are interested in. For example, in the case of birds, in moderate disturbed areas, there may be more species might be there, whereas in the undisturbed areas there may be less species. It is because of the difference in the niches and in the difference of the habitat use of the animal. So it's a good question, but it's much more than what it means. So because we have not talked about species richness in this talk at all, but broadly I can say that uh, how to estimate species richness itself is a big topic, because it is not just that you in a area you simply go and look at the number of species. So there are several methods are there. So uh, it's a different topic. So I would just like to say that this particularly we are now talking please do not get confused between population density, abundance, and species richness.
Thank you very much, sir. Hope the Netravati has received her answer. If any other questions or participants would like to ask any questions to sir, they can raise their virtual hand or mention in the chat box. Indeed, in fact, sir, wonderful presentation, and that video was very helpful. That we came to know that how the count of the animals has been done in the forest area. It gave us a glimpse of knowledge, and we came to know about the sampling methods also. Thank you very much, sir, for that.
Okay,
In
welcome.
fact, yes, sir.
Anytime that you have my email ID that can be distributed to all the participants, and if they have any questions, they can write to me by email also.
Sure, sir. Sure, sir. In fact, my camera is not working, and I thought we can invite you, sir, in person to our academy office also, so that we can
Okay.
give some presentation to interested people and general public regarding the same thing. It will
Oh
be
yes,
helpful.
sure. I will be happy to do that whenever time permits. Yeah, sure.
Sure, sir. Sure, sir. Thank you, sir. Meanwhile, I request
Okay.
once again, if any other questions, participants can write to sir or contact us. We will get back to you with the answers. Uh, Siddhegaudre, shall we wind up? Okay, sir. Now, formally, I would like to
Okay.
thank you once again on behalf of KST, on behalf of all the teachers and the students, and on my own behalf, sir. Wholehearted thanks to you for accepting and delivering today's session. Thank you very much, sir.
Okay.
Also, I would like
Thank
to
you.
thank our KST staff, technical team, non-technical team, and teachers and students who are with us today in this platform as well as in the YouTube channel. Thank you. See you
Okay.
Monday. Thank you. Thank you, sir.
Right. Thank you.
Thank you, sir. Thank you very much, sir.